Welcome to Get Moving TV. I am always amazed at what happens here in Ventura County. You hear the Silicon Valley, we're like a gallium gulch out here. Today we're going to have two big technical innovations that have occurred here in Ventura County. And we're going to talk about the applications that we're working on to really apply them to healthcare, to improve the health of children with cystic fibrosis, improve the children's health of children with asthma, and really get an idea about how to put this technology to work. My first guest today is Dr. Ed Staples. Ed and I became acquainted when I heard about his product and thought maybe there's a way we could apply it to cystic fibrosis. Ed, could you tell me a little bit about yourself and the company you founded?
Yes, I'd be happy to, Chris. I've been involved in the development of electronic noses for about 20 years. And there's a number of researchers around the world who've been working these issues. In about 1995, I invented an electronic nose based on high-speed gas chromatography. We founded a company called Electronic Sensor Technology. That company today manufactures an electronic nose called Z-nose, or we like to say the Italian pronunciation Z-nose. More recently, I formed a second company working on solutions or applications of this electronic nose technology. You mentioned a few already, which we consider in the life science area, the smell of living beings.
Well, tell me, I know there are 5 major uses for this device. What are the major areas where?
This is one of the most exciting technologies that I've ever been associated with. There are so many applications. We like to break them down into 5 major areas. The life science, which is the smell of living beings, which is really a biochemistry, a chemical petrochemical, which encompasses a very broad spectrum of chemical products in general. Environmental, which implies air, water, and soil, looking for contamination or pollution. Food and beverage, one of my favorites. This technology is excellent for looking at quality control, food quality, as well as food safety, which gets back into a little bit of the life science with the bacteria contamination. And finally, last but not least, is security, looking for explosives, looking for contraband, drugs, all kinds of applications in that area, particularly today with the accent on terrorism and the vast, very wide proliferation of different types of terrorist threats.
Well, here in Ventura County, we have California State University Channel Islands, and along the coast, Cal Poly San Luis Obispo. And we've really tried hard to get the faculty from those universities and the college involved. What's the project you're working on now up at Cal Poly?
We have a very interesting project going on at Cal Poly aimed at the Salinas Valley and the Santa Maria Valley produce production. You may have, and I'm sure you have recently in the news, the problems associated with E. coli or fecal contamination of produce such as lettuce, broccoli, and spinach. So we've got a project with the Cal Poly researchers, Professor Kitts, and we're attempting to screen the produce coming literally from the field into the packing plants, looking for the presence of fecal contamination, E. coli, bacteria, anything out of the ordinary.
What a great innovation. Certainly we've seen the bad effects in our children, and so something that could be used at the level of the factory or even the supermarket.
Yeah, these types of bacteria particularly affect the young and the old, and in fact can be quite deadly.
Now, Homeland Defense, I know the company had run off in that direction. How is it used in Homeland Defense?
Primarily looking for explosives. We actually have a number of units right now in Iraq that are being used to try to combat the improvised explosive devices, which are killing so many of our boys over there.
Could you describe a little bit about how the machine works itself? And
certainly, it's all about early electronic noses were based on pattern recognition, and they gave a rather vague pattern, but it could be recognized. But they didn't separate the chemicals. They weren't able to tell you the individual chemicals. And so a mixture of different odors would confuse the machines, and they had some real difficulties. Later in the late 1980s, two groups, one myself here in Southern California, another one in Toulouse, France, developed electronic noses based on gas chromatography. Gas chromatography is one of the oldest sciences around. It's over 100 years old. It's primarily in laboratories, and it tends to be very slow. And as you know in your practice, you take samples, you send them to the lab, and you wait sometimes days to get the results. What we did was solve the problem and make it fast. We now can do gas chromatography in 10 seconds, in real time, portable instruments, battery operated. So really this is a tremendous opportunity for getting instruments at the POC or point of care, where right there with the patient you can do a screening. They're not foolproof, so it is a screening technique.
All right, Ed, well, let's take a look at what you've brought. I see we have a mask there. What's this for?
Yeah, Chris, this is what we're using to sample air from children from their breath. We actually attach this with a outlet here where they exhale through this. It goes into the machine, or we can collect it into a nylon bag, and then we pull air out of the nylon bag, which came from the breath, into the Xenos, and the Xenos analyzes the chemicals that are in the breath.
I hear the machine's making some noise now. Can we kind of run through a sample?
Yeah, the way the instrument works, it's a two-cycle. Initially, it breathes air in, and that's what you hear, that little humming noise is a pump, which is sucking air in. We can control how much sample we suck in. We take all the chemicals that are in that sample of air, concentrate it, and then we analyze. And the following sampling takes about 10 seconds, and we analyze the chemicals, and we'll see a spectrum of light chemicals, medium chemicals, to heavy chemicals. And I have a device here which we use to calibrate the instrument, which can very easily show you how it works. The machine is operating right now on an automatic cycle, and it'll come around and it will start to operate again. And while it's sampling, I'm going to give it a short little burst of chemicals, which are a series of what's called N-alkanes. And you'll see the spectrum appear after it samples in about 10 seconds.
We were talking a little bit about how the dogs could smell the difference between humans and the Terminator in the movie The Terminator there. How sensitive is this?
This device is comparable to a dog in sensitivity.
So much better than humans.
Now you hear the sampling. I'm going to just give it a quick little squirt, and you'll see a spectrum here when it's done sampling. And now it's analyzing.
Well, that's pretty amazing, the speed at which all that occurs. Are there fingerprints, kind of vapoprints that are typical for different bacteria or other compounds?
Yes, there are. We can actually define the chemicals associated with different kinds of bacteria, and we can set up alarms. So instead of looking at a spectrum, which is what we have here, we can actually show a bar display like this here, which shows, in this case, 7 primary chemicals that we're searching for. So that when the instrument runs, we'll basically look for red light, green light, and we can set the threshold of these sensors. So we can determine, for example, when we're looking for E. coli on lettuce, we're looking for a chemical called indole. When we're looking for cystic fibrosis infections in the lung, we're looking for a ketone called nonanone, as well as phenol and some others. These compounds are called biomarkers, and they uniquely identify that particular bacteria.
Well, Ed, thank you very much. This has been an excellent demonstration of the kind of technology we have here in Ventura. It's the kind of thing that just keeps us moving.